So, hi everyone. Today is the 8th problem solving session for this course, Biochemistry. The course instructor, as you know, is Professor Swagata Dasgupta from IIT Kharagpur, and I am Shrija Ghosh, a PMRF from IIT Kanpur. I'll be helping you solve all the assignment questions that are there for this week's topic. The topic is nucleic acids. So, we'll discuss different questions about nucleic acids, and then you can ask me any doubt if you have. Okay, so you can either ask me your doubts by unmuting yourself, or you can write your questions in the chat box. Okay, whatever is comfortable to you. So we'll start. Just a second. Yeah. So we'll start with the questions. Feel free to stop me whenever you have any doubt. Okay, so starting with the first question, we have a diagram here. Identify the overall process being depicted in the provided diagram. So what is the overall process that has been depicted in this diagram? So in this diagram we can see there's a DNA molecule that is going to RNA molecule, and then that RNA is getting converted into protein. So what is this overall process called? Option A, transcription. Option B, central dogma of biology. Option C, replication. And option D, transmission. So what is this process called? People have answered option B, central dogma of biology. Very good. So this whole process where the DNA gets converted into RNA and then finally into protein, this is the central dogma of biology. Why the central dogma? Why it is named as central dogma? It is basically the process that is revolving around the life processes that are happening on happening around yourself. So for any function to happen, you need a protein, you need a machine that can drive that function, right? For that protein to form, you need an RNA molecule, and for that RNA to form, you need the DNA molecule. So all the information that is there, that has to be done by your body, all that information is written in forms of codes that are basically called genes. Okay, and these genes are basically part of DNA molecules. So these parts of DNA, each gene gets converted into an RNA molecule, and that RNA molecule then makes that particular protein that is coded by that particular gene. Okay, so this is a central dogma of biology. This is the most basic process around which life evolves. Okay, so this is the central dogma of biology. Second question, now identify the processes X and Y that results in the formation of RNA and protein. So you have to tell me the name of the process by which DNA is converted into RNA, and then the process by which RNA is converted into protein. So what would be the answer? Okay, Akshara has answered option A. X is transcription, Y is translation. Yes, very good. So that means the process of conversion of DNA into RNA, this is called transcription, and the process of RNA getting converted into protein, this is called translation. So you have a gene. Where is the gene present? Gene is present in the DNA, right? So that gene will code for a protein, but for that protein to be formed, first mRNA will form from that gene by a process called transcription. Okay, and then that mRNA will get converted into protein that is basically coded by this gene, right? So from gene to protein, the process will go through transcription, and translation. Okay, so a DNA is always transcribed into mRNA. So you know DNA and RNA both are what? Nucleic acids, right? This Na is nucleic acid, this is deoxyribose nucleic acid, and this is ribose nucleic acid. So from nucleic acid, from one form of nucleic acid to another form of nucleic acid, the process is transcription, because it's basically copy of one type of molecule into a variant of that molecule, right? But in this case, now RNA is getting converted into protein. These are biochemically different molecules, right? This is a nucleic acid and this is a protein. What is protein? Protein is made up of amino acids, right? So this is a chain of amino acid. Here this is chain of nucleotides. So now this is being translated into some other biomolecule. So this process is translation, whereas this process is transcription. Okay, I hope this is clear. Okay, moving to the next question. Question number 3. Which of the following options does not correspond to the pyrimidine bases found in RNA? Okay, does not correspond. First is uracil and thymine, second is thymine and cytosine, third is adenine and thymine, and fourth is cytosine and uracil. So which of the following options does not correspond to pyrimidine bases found in RNA? What would be the answer? Which bases are not found in RNA? Yeah, tell me. Option C. Yeah, okay. Option C means 2 and 3. So thymine and cytosine and adenine and thymine. Why not why not the first statement, uracil and thymine? Thymine is not present in RNA, right? So the correct option is option D. So you know RNA and DNA, the main difference between the bases that are found in RNA and DNA is, in RNA we have base uracil, whereas in DNA in place of uracil we have thymine. So in DNA we have ATGC and in RNA we have AUGC. Okay, so in place of thymine we have uracil in RNA. So all these options have thymine in them. So we can have uracil in RNA, but we cannot have thymine. Again, we cannot have thymine, we can have cytosine. We can have adenine, but we cannot have thymine. Only in this case we can have both cytosine and uracil in the RNA. So which of the following options does not correspond to pyrimidine bases? That is all first, second and third. All these cannot be there in the RNA bases, only cytosine and uracil. Okay, C and U. Okay, is this clear why option D is correct and not C? Okay, yeah, okay. So moving to the next question. Most of you had answered option C, so I hope it's clear to everyone why option D was correct in that question, right? Okay. Question number 4. Identify the correct statement regarding the nucleic acid structures. First statement is nucleic acids are made up of polymers of 4 different nucleotide residues each. Amongst these, RNA uses AMP, TMP, GMP and CMP. DNA uses the deoxy forms of deoxy forms, that are DAMP, DCMP, DGMP and DTMP. So identify the correct statements regarding the nucleic acid structure. So tell me which of the following 3 statements are correct. So first statement says nucleic acids are made up of polymers of 4 different nucleotide residues. Second statement says RNA has AMP, TMP, GMP, CMP, and third statement is DNA has DAMP, DCMP, DGMP and DTMP. Which of the following statements are correct? Okay, we have option C. Okay, my voice is echoing. Is this a problem for everybody, or if only Neeta you are facing this problem, I would suggest you to disconnect and rejoin, maybe. Are you also facing this problem? Akshara, can you respond? Okay, then Neeta, you should disconnect and rejoin. Yeah, others are not facing this issue, so just try disconnecting and rejoining. It would help you. Yeah. Okay, so Akshara had answered option C for this, right? That means all 3 statements are correct. Okay. Anyone else wants to answer? What would be the correct answer for this? Okay. So, first statement is definitely correct. Nucleic acids are made up of 4 different nucleotides. So, I told you they are made up of A, T, G, C, and in RNA in place of T we have U. So, second statement, if you see among these, RNA uses AMP, TMP, GMP and CMP. So, RNA does not have TMP. In the previous question only we saw in RNA we have uracil in place of thymine. Okay, so in DNA we have ATGC, in RNA we have AUGC. So, the the monomer of RMP would be UMP. Why? What is M and P in this? It is basically monophosphate. So, these are basically the monomers of the polymer, that is the nucleic acid. Okay, so RNA is made up of AMP, UMP, GMP and CMP. And in case of DNA, the only difference between DNA and RNA is they have the 2' hydroxyl group removed. So, this is deoxy AMP, deoxy CMP, deoxy GMP and deoxy TMP. Okay, so the correct answer is first and third statements are correct. That is option D. Option D is the correct answer. Okay. Yeah, Neeta had answered option D. First and third statements are correct. Okay. So, you see this is one nucleotide and this is another nucleotide. Can you name these nucleotides? What is the name of this nucleotide and this one? Let's say first and second. So, what is the name of the first nucleotide? Anybody? So this is AMP. So you see this here, this molecule here, this is nothing but adenine. Okay, this is adenine. It is attached to a ribose sugar and it has a single phosphate attached. Okay, so it is called adenine monophosphate and it has ribose with both the OH attached. So this is the first carbon, this is the second carbon, this is the third carbon, this is the fourth carbon, and this is the 5 carbon. It's a 5 carbon sugar. Ribose is a 5 carbon sugar. So you see in on the second and third carbon OH are attached. So this is a ribose phosphate, adenine, ribose, monophosphate. Okay, so this is a monomer of a RNA molecule. Okay, and here you see what is the difference between this and this structure, first and second structure. If you see, the only difference is this OH at the second carbon position is missing. So this is the first carbon, second carbon, third carbon, fourth carbon and fifth carbon. Again this is the adenine molecule, this is the this is called deoxyribose. Okay, this is called deoxyribose. Why deoxy? Because one hydroxyl group is removed from this. So this group, this is not there in this, so it is called deoxyribose. So deoxyribose sugar is there and adenine is there and a phosphate group. So this is called DAMP, and this is a monomer of a DNA molecule. So DNA, deoxyribose nucleic acid, and RNA is ribose nucleic acid. This is clear? So that's why you have, for RNA you have the monomers written as AMP, CMP, GMP, and the fourth monomer is UMP in RNA. In DNA you will have DAMP, DCMP, DGMP and DTMP. In DNA you will never have uracil, in RNA you will never have thymine. Okay, this is the structure of, I'll just omit this, just a second. So this is the structure of uracil and thymine, you can see here. So the only difference here is the methyl group. So in uracil you don't have the methyl group, and in thymine you have an additional methyl group. So this thymine is present in DNA and this uracil is present in RNA molecules. Okay, both are pyrimidines. The only difference is the methyl group on one of the carbons. So this is present in DNA and this is present in RNA. Okay, I hope this is clear. So that's why you had in this question the second statement as the wrong statement. Both the other statements that are given are correct. Okay. Okay. Moving to the next question, question number 5. What is one of the primary differences between purines and pyrimidines of DNA? So we have been talking about purines and pyrimidines, the monomers of DNA, this nucleic acid. So what is the difference between purines and pyrimidines? Option A, purines consists of single ring whereas pyrimidines consists of double ring. Option B, purines have a lipid chain whereas pyrimidines do not. Option C, purines consists of 2 rings whereas pyrimidines consists of 1 ring. And option D, purines bond to phosphates on the DNA backbone whereas pyrimidines bonds to sugar. What would be the correct answer here? What would be the correct answer? The correct answer is option C. Purines have 2 rings and pyrimidines have 1 ring. So that is the major difference between purines and pyrimidines, the structure. Okay, so we will see the structure here. So you see we have purines with 2 rings attached. So one 6 carbon ring is fused with a 5 carbon ring, and here we have a pyrimidine which is a 6 atom ring. So this is pyrimidine and this is purine. So we have, I told you we have 4 bases A, T, G, C, right? So among that, A and G, adenine and guanine, are purines, and C, T and U, that is cytosine, thymine and uracil, are pyrimidines. Okay, so modification on these side chains, basically on any of these atoms, will give rise to adenine or guanine, and modification in any of these side chains, these atoms basically will give you cytosine, thymine and uracil. So this is a purine molecule, this is a pyrimidine molecule. The difference is basically in their structure. Okay. So next question, identify the structures X, Y and Z. So we have 3 structures given here, X, Y and Z. You have to name what is X, what is Y and what is Z. Option A, X is a nucleobase, adenine, Y is a nucleoside, 2-deoxyadenosine, Z is a nucleotide, 2-deoxyadenosine, 5' monophosphate. B is, X is nucleoside adenine, Y is nucleotide 2-deoxyadenosine, and then Z is nucleobase 2'-deoxyadenosine 5' monophosphate. C says X is nucleobase, Y is nucleotide, Z is nucleoside. Then D says X is nucleotide, Y is nucleoside and Z is nucleobase. So what would be the correct answer here? Yeah. Okay, Neeta says and Akshara says option A. Yes, very good. Option A is the correct answer here. So basically you see this is again an adenine molecule. This is what? This is a purine. I showed you in the previous slide, purine is a 2-ring molecule. So we have a amino group attached to one of the carbons in this purine base structure, which makes it adenine. So it's a single base. This is called the bases of DNA or bases of nucleic acid. So this is a nucleobase, base of a nucleic acid, nucleobase. So X is a nucleobase. When this base is attached to a sugar molecule, so this is a deoxyribose sugar. Why? This you can see on the second carbon, instead of OH only H is there. So this is a deoxyribose. So to this deoxyribose, when a base is attached to the first carbon, this makes it a nucleoside. Okay, nucleoside means sugar plus base. Okay, only base is called nucleobase. When the base is attached to a sugar, it's a nucleoside, and the name is given as 2'-deoxyadenosine. Why 2'-deoxyadenosine? So 2'-deoxy suggests that the second carbon. So I told you this is the first carbon, this is the second carbon, third carbon, fourth carbon and fifth carbon, right? So second carbon the oxygen is removed. So that's why 2' deoxy. And adenosine means the adenine base is attached to a ribose sugar, making it adenosine. Okay, so this is a nucleoside. Now in the third structure, you see what is the difference between the second and the third structure? This phosphate is added. So now we have a base, we have a sugar, and we have a phosphate. Okay, and from to which carbon the phosphate is added? This is the fifth carbon, right? Remember, this is the first carbon, this is the second carbon, third carbon, fourth carbon, and fifth carbon. To the fifth carbon, phosphate is added, and this is called a nucleotide. Nucleotide has a base, a sugar, and a phosphate. So only base is nucleobase, base plus sugar is nucleoside, and base, sugar, and phosphate is nucleotide. In this nucleotide, now the naming goes as 2' deoxy. This I have explained why 2' deoxy. The second carbon does not have the oxygen, so 2' deoxyribose. So 2' deoxy. Adenosine. Adenosine means adenine is attached to a ribose, so adenosine. 5' monophosphate. 5' monophosphate. Why? With the fifth carbon, phosphate, only single phosphate is attached, so this is monophosphate. If I attach another phosphate here, it will become a diphosphate, triphosphate, like that. Okay, so 5' monophosphate means single phosphate is attached, okay, to the fifth carbon. So second carbon deoxy, fifth carbon monophosphate. This is clear. So this is a nucleotide. So you have only base, that is nucleobase. You have base plus sugar, nucleoside, and base plus sugar plus phosphate, nucleotide. This is clear. So correct answer is option A. Now we move to the next question, question number 7. Which among the following are not the feature of Watson-Crick pairing? Which among the following are not the feature of Watson-Crick pairing? First statement is the permitted hydrogen bonds are adenine with thymine with 2 bonds and guanine with cytosine with 3 bonds. So this is the number of hydrogen bonds given. Adenine and thymine will have 2 hydrogen bonds and guanine and cytosine will have 3. Second statement is the Watson-Crick base pair is a non-planar structure. Third statement says purine and pyrimidine bases are saturated with hydrogen bonds. And the fourth statement says the beta glycosidic bond is attached on the same edge of the base pair. So what would be the correct answer here? Which of the following statements are not the features of Watson-Crick pairing? So you have to find the incorrect statements. Okay, Neeta says option D, that is the fourth statement only. Okay, anyone else? Okay, we have option D here. Let's see. So you say fourth statement is not the feature of Watson-Crick pairing. So now you see, first statement we have hydrogen bond between adenine and thymine and guanine and cytosine. So this is a correct statement. Watson-Crick pair states that adenine will form 2 hydrogen bond with thymine and guanine will form 3 hydrogen bonds with cytosine. So this is the first Watson-Crick pair rule. So this is a Watson-Crick pair where adenine base pairs with thymine with 2 hydrogen bonds and guanine base pairs with cytosine with 3 hydrogen bonds. So this is a correct statement. Okay. Now the second statement where they say that the Watson-Crick base pair is a non-planar structure. You see, it's an incorrect statement because when the base pairing is happening, so when the base pairing is happening, the bases are actually lying on a plane, and the sugar phosphate backbone, the phosphate groups will protrude in and out of the plane. Okay, so when you have a chain of a nucleic acid, like the whole polymer, you will have a sugar attached to a phosphate and a base. This base will base pair with another molecule, right? So you will have here something like this. You will have your sugar attached to a phosphate and the base, right? This base will base pair with another DNA molecule. You will have the sugar and the phosphate. So this phosphate group will, this sugar and phosphate group will go in and out of the plane, and this base pair will be on a plane. So this will be a planar structure. So Watson-Crick base pair is a non-planar structure. This is not the feature of Watson-Crick pairing. This is an incorrect statement. So second statement is incorrect. Now purine and pyrimidine bases are saturated with hydrogen bond. This is also actually an incorrect statement, because if you see the structure of purine or a pyrimidine, they have other atoms that can form hydrogen bonds. So it is not completely saturated with hydrogen bonds. So this is also an incorrect statement. The fourth statement, beta glycosidic bond is attached to the attached on the same edge of the base pair, that is actually the correct statement. Why? What is the beta glycosidic bond here? The beta glycosidic bond is the bond between the sugar and the base. So if you see, if you have a DNA molecule, a double stranded DNA molecule like this, the beta glycosidic bond, that is this bond, will be on the same edge of the base pair. If this is the base pair edge, this bond will be on the same edge, right? So the second and third statement are incorrect. So option C is the correct answer here. Okay. So this is actually how base pairing is happening. So this is the adenine molecule and this is a thymine molecule. So you see in thymine, this O can also form a hydrogen bond. So this is not completely saturated. So that's why we had purine and pyrimidine bases are saturated with hydrogen bonds, this as the incorrect statement. Okay. So this is making a planar structure, not saturated with hydrogen bonds, and the beta glycosidic linkage, that is this bond, it is on the same edge as the base pair. So this and this are on the same edge as the base pair. So base pair is here, it is on that same edge, it is not on the opposite side. Okay, here also this bond and this bond are the beta glycosidic linkages. Basically the bond that is formed between the bases and the sugar. So here with what we have the sugar, right? Deoxyribose in case of DNA and ribose in case of RNA. Here also we have the sugar, right? So this is the beta glycosidic linkage. So they are on the same edge as the base pair. And you see 2 bonds are formed between adenine and thymine and 3 bonds are formed between guanine and cytosine. What are these bonds called? These are hydrogen bonds. Okay, these are hydrogen bonds that are formed between the different bases, purines and pyrimidines. So you see double ring structure, this is a purine forming hydrogen bond with a pyrimidine. Again, this is a double ring structure, purine forming a hydrogen bond with pyrimidine. Okay. So moving to the next question, which of the following statement is not true regarding the double helix and the features of Watson-Crick pairing? First statement is the combination of the staircase sugar phosphate backbone and the bonding between the base pairs results in the double helix. Second statement is 5' 3' strand is known as sense strand, whereas 3' 5' strand is known as antisense strand. third statement is dimensions of the two permitted base pairs in the Watson-Crick pairing are similar. And the fourth statement is the original Watson-Crick model was based on the aDNA, which is predominant form under the physiological conditions and has 10.5 bases per turn. So, which of the following is incorrect? Which of the following is incorrect? Come on. Sorry, your voice was not clear. Okay, Akshara says option D. Yeah, so, sorry. Yeah, the correct answer is option D, where the original Watson-Crick model was actually based on the bDNA. Okay, it was based on bDNA, not the aDNA. We'll see in further slides there are three types of DNA: aDNA, bDNA and zDNA. So, the original model was based on bDNA, it was not based on aDNA. Rest all the statements are correct. The DNA forms a double helix because of the sugar phosphate backbone, and then the bases making hydrogen bonds, bases pairing with each other, forming hydrogen bonds. Then we have 5' 3' strand known as sense strand, and 3' 5' strand known as antisense strand. This is also there. Why? From where we are getting this 5' and 3'? I told you the fifth carbon that is attached to the phosphate. From that, whenever we are seeing a DNA strand, whenever we are naming the DNA strand, we always start from 5' to 3'. So that is the sense strand, that is the forward strand, and then the reverse strand will be 3' to 5'. Why the name sense and antisense comes? So basically, whenever from a DNA molecule, so you saw in the central dogma, you had DNA that was going to RNA, right? So the DNA is a double stranded molecule, which will have a 5' to 3' and 3' to 5', but a RNA will have only a single strand with 5' to 3'. Okay, so basically whatever the template for this DNA for this RNA molecule, the template will be only one strand of the DNA molecule. So that means the strand that is the template is the sense strand, and the opposite strand is the antisense strand. Okay, then you have dimensions of the two permitted base pairs in the Watson-Crick pairing are similar. So that is true. Then only you can have a planar structure for the base pair, and then the helical structure for the complete DNA. And then the original model is based on aDNA, which is incorrect. It was based on bDNA. Okay, so the correct option is correct answer is option D. Now moving to the next question, question number 9. Rate of hydrolysis is slower in DNA than RNA. This is because option A, the ribose sugar present in DNA is in deoxy form, but is not so in RNA. Option B, this is because of the presence of a different pyrimidine in RNA compared to DNA. Option C, this is because of the stacking of nitrogen bases. And option D, this is because of the phosphate in the backbone of RNA favors hydrolysis at a faster rate. So what would be the correct answer? Why rate of hydrolysis is slower in DNA than RNA? What makes RNA very unstable? Yes, so okay, we have option A as the answer, which is correct. Option A says ribose sugar present in DNA is in deoxy form, but is not so in RNA. So if you remember from the previous session, we had discussed about RNA hydrolysis by enzyme called ribonuclease. So in that mechanism, or in general acid base hydrolysis of RNA, what happens is the 2' OH that is there, the hydroxyl group, it gets deprotonated, and then this oxygen can easily attack this phosphate that is there in the RNA attached to the 3' carbon, and then the hydrolysis is favored. In case of DNA, since that 2' OH is not there, so this attack is not favorable for DNA to get hydrolyzed. An external water molecule has to get deprotonated, and an external OH minus ion has to attack this phosphate. So that's why RNA molecule is unstable, because it can get auto hydrolyzed. Sorry. So in an RNA molecule, it can get auto hydrolyzed because it has a OH group that can easily get deprotonated. So under alkali conditions, what will happen? The deprotonation will happen if we increase the pH. All the protons will be lost in the solution in order to neutralize the solution. So the OH, as soon as the proton is lost from the OH, we will have a free radical or a O minus ion. O minus can now attack that phosphate. So if a molecule has its own O minus, it is more likely to attack itself than an external O minus or OH minus ion to come and attack a molecule. Okay, so in case of DNA, an external OH minus has to come and attack, but in case of RNA, it itself has that O minus ion to attack its phosphate group and hydrolyze the RNA molecule. Okay, so that's why DNA hydrolysis is slower. DNA is more stable structure than RNA, and that is actually one of the major reasons why DNA is our genetic material and not RNA. So earlier, when the discovery of nucleic acids were being made, so at that time the question was whether DNA was the one that is carrying our information or RNA. So this is one of the primary reason why DNA, by nature, might have selected DNA to be the genetic material, because DNA is a more stable molecule than RNA. Okay. So moving to the next question, question number 10. 3 different segments of A DNA, B DNA and Z DNA all contain 1320 base pairs. What is the approximate length of the longest DNA possible? So first we have to think which of the following A DNA, B DNA and Z DNA is the longest DNA, and then we will calculate. Okay, so I told you there are 3 types of DNA. Okay, A DNA, B DNA and Z DNA. What is the difference between all 3 types? All are helical structure, all are double stranded. The difference is in the length of that helix. So the difference is how many number of bases are there per turn of helix. So when you were studying the helical structure of proteins also, you remember in alpha helix you had particular number of residues per turn, and then the length of each turn was defined. So similarly for DNA also, you see here, this is the characteristics of A DNA, B DNA and Z DNA. So the pitch means the distance between each turn. So let's say this is a helix. Okay, so this here is one turn, right? So the distance between each turn, this distance between each turn, this is called pitch. So for A DNA, you see the pitch is smaller, 2.8 nanometer. For B DNA, it is similar to our alpha helix, it is 3.4 nanometer, and for Z DNA, it is higher, 4.5 nanometer. So you can see, if this length, this distance is more, that means the molecule will be more stretched out. That means you will have the longest DNA to be Z DNA. So it is asked in the question, what is the approximate length of the longest DNA possible? So longest DNA would naturally be Z DNA because it is more stretched out. So it would be longer than A and B. The shortest will be A DNA here. Even if you see the number of base pairs per turn, so in one turn, so this is one turn, right? This is one turn. So in one turn, you will have 11 bases in case of A DNA, 10 in case of B DNA, and 12 in case of Z DNA. Naturally, the turn is also higher, number of bases is also higher. That means Z DNA is the longest DNA. Okay. So now we will do the calculation based on the Z DNA. Okay. The longest DNA, that is Z DNA. We will do the calculations based on that. So we have in the question, number of base pairs given as 1320. So how will we do the calculation? How will we do the calculation? Yanita is saying Z is left-handed. Yes, that is also a major difference between BDNA and ZDNA. BDNA, the DNA form that is most commonly present, is a right-handed helix, but ZDNA is a left-handed helix. Okay. So to calculate this, we have 1320 base pairs, and it is given that in one turn we have 12 base pairs. So how do we calculate how many turns are there? So to calculate number of turns, we will basically divide 1320 by 12. So in one turn we have 12. So how many turns will be there in 1320? First we will calculate that, and then we will calculate. So this is the number of turns. Okay, 110 is the number of turns. Now length will be, for one turn the length is 4.5 nanometer. So for 110 turns, what would be the length? It would be 110 into 4.5 nanometer, that is 495 nanometer. So if you see the options, option A is the correct answer. 4.5 nanometer, 495 nanometer is the length of the longest DNA possible. If I would have asked you to calculate the length of B DNA formed by these many base pairs, you would just have to divide it by 10. That would give you 132, and then 132 into 3.4 would be the length of B DNA with 1320 base pairs. Okay. Similarly, you can calculate the length of any of the forms of DNA if you have the base pairs given. Okay. Total number of base pairs can help you find the length of the DNA if you know the form of the DNA. Okay. I hope that is clear. So moving to the next question. Previous question was clear to everyone, right? Okay, so we'll move to the next question, question number 11. Which are the correct statements regarding exo and endonucleases? Exonucleases catalyze the removal of terminal nucleotides 3' and 5' types. Second statement is endonucleases catalyze the removal of external phosphodiester bonds. Third statement is type A endonuclease acts on the 3' hydroxyl of the nucleotide with the phosphorus group. And fourth statement is type B exonucleases act on the 5' hydroxyl group of the nucleotide with the phosphorus group. So, which are the correct statement regarding exo and endonucleases? Anyone wants to answer? Okay, so option D. Option D means first, third and fourth statement are correct. Okay, so first statement, exonucleases catalyzes the removal of terminal nucleotides 3' and 5' tribes. Yes, that is correct answer. Exo means outside, endo means inside. So, exo cuts the ends of the DNA, whereas endo cuts in between the DNA molecules. So, endo catalyzes the removal of internal phosphodiester bonds. Okay, it removes the internal phosphodiester bonds. Then type A endonucleases acts on 3' hydroxyl of the nucleotide. So, that is also correct. Type A. So, we have two types of endonucleases, type A and type B. So, type A cuts the 3' hydroxyl group of the nucleotide that is present inside, and type B, if you pay attention, here we have written type B exonucleases. So, this is also an incorrect statement. Type B endonucleases acts on the 5' hydroxyl group. Okay. So, only first and the third statement are correct. Fourth statement is not correct, because you see here this is exonucleases, but the endonucleases actually acts on the 5' hydroxyl group of the nucleotide with the phosphorus group. Okay. So, the correct answer would be option C, not option D. Fourth statement is wrong. Okay. So, you have to pay attention to what is written carefully, because these small one word change can actually change the whole meaning of the statement. Okay. So, option C. So, here you see exonucleases cuts the outer part 5' and 3' ends, whereas endonucleases are cutting in between. So, that is internal cuts, internal phosphodiester bonds. These are external cuts. So, exo, internal, so endo. And it says type A and type B. So, basically, if you have a 5' to 3' group, so you will have a 5' phosphate and then a sugar and then a base attached to it. Then from the sugar again a phosphate will be there, another sugar will be there and a base attached to it, and so on and so forth. So, the cutting that is happening, it is at the 3' hydroxyl group of the nucleotide. So, this is one nucleotide, this is the second nucleotide, then again you will have a phosphate and a sugar. So, this is the third nucleotide. So, if this is the 5' end, this will be the 3' end, again this will be the 5' end, this will be the 3' end, right. So, type A basically cuts at the 3' end of the hydroxyl group of a nucleotide. So, if it is cutting at this nucleotide, it will cut here, okay, at the 3' end, and type B will cut at the 5' end. So, this will be type A, this will be type B. Okay. So, option C is the correct answer. Type B endonuclease will cut at the 5' end and type A endonuclease will cut at the 3' end. This will exonucleases always cut at the ends of the DNA. So, you see in this figure, exonucleases will cut at the ends of the DNA, and endonucleases will cut in the internal phosphodiester bonds. Okay. So the next question, question number 12. What should be the complementary strand of the given DNA sequence? So it is given as 5 prime T A C G G A A C T 3 prime. Okay. So what would be the complementary strand to this? This is 5 prime to 3 prime. So I told you A base pairs with T and G base pairs with C. So you take your time and tell me the answer. What would be the correct answer? Okay, Neeta says option D. Okay. 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 So let's see. Akshara also says option D. So let's see whether it is option D or not. So we have, so first thing you have to pay attention is the prime that is given. So opposite to 5 prime it will always be 3 prime. Okay, and opposite to 3 prime it will always be 5 prime. Now it will be 3 prime. Opposite to T we will have A. Opposite to A we will have T. Opposite to C we will have G. Opposite to G we will have C. Again a C. Then A will give T. Again a T. C will pair with G, and then again an A, and then we will have opposite to 3 prime we will have a 5 prime. So the sequence should be A T G C C T T G and A, 3 prime to 5 prime. Okay. So yes, option D is the correct answer. So sometimes what will happen in the options, if the option was written from this direction to this direction. So what if the option had 5 prime A G T T C C G T A 3 prime. So in that case you will have to be more careful. So instead of this option, if I would have given you this option, then also you will have to pay attention what is the ends that are written here. So if you are reading from 5 prime, you will have to see that okay, 5 prime will pair with 3 prime. So this end will be opposite to this base pair, and then it will go in this direction. Okay, for this the base pairing will go in this direction. Okay, so we can write this strand in both ways, but while finding the complementary strand to a given strand, you will have to make sure you are reading it in the correct direction. Okay, so option D is the correct answer. Okay. So moving to the next question, which among the following statement is or are false about the stability of DNA? Okay, so we have 4 statements here. First statement is charged and hydrophilic sugar phosphate backbone are in the interior. Second statement is stabilization through base pair hydrogen bonding is greater than the stacking interactions. Third statement is hydrogen bonding guarantees the complementarity of the strands. And fourth statement is stacking interactions are sufficient to overcome the electrostatic repulsions of the phosphate. So which of the following are false about DNA stability? Tell me. Anybody? Which of the following statements are false? Charge and hydrophilic sugar phosphate backbone are in the interior. Is it true or false? Stabilization through base pair hydrogen bonding is greater than the stacking interaction. Hydrogen bonding guarantees the complementarity of the strands. and stacking interactions are sufficient to overcome electrostatic repulsions of the phosphates. Which statements are false? So the correct answer is option A. Except the third statement, all the other statements are false. So first statement goes like charged and hydrophilic sugar phosphate backbones are in the interior. Now if you remember about polarity, polar and non-polar, I told you in case of proteins also we had all the polar and charged groups towards the outer side, and what was in the interior, the hydrophobic groups, right? So remember in the proteins also you had charged amino acids on the outer side and the hydrophobic amino acids on the inner core. Similarly, this is true for all biomolecules. For membranes also you saw polar head groups were on the outer side and the non-polar tails were towards the interior of the membrane. So this is actually true for all the biomolecules. So for DNA also it's another biomolecule. So DNA also will have the charged and hydrophilic moieties towards the exterior, and towards the interior what will be there? Interior will have the bases that are hydrophobic. Okay, bases will be hydrophobic, so they will be there in towards the interior, and towards the exterior we will have the charged and hydrophilic sugar phosphate backbones. Okay, so first statement is false. Second statement, stabilization through base pair hydrogen bonding is greater than the stacking interaction. This is also false. Actually, the base pair hydrogen bonding, the stability provided by the base pair hydrogen bonding is almost equal to the stability provided by the stacking interaction. So all the whole DNA molecule is stable because of both base pair hydrogen bonding and the stacking interaction. So all the sugar phosphate backbones are stacked upon each other, and the bases are paired with hydrogen bonding. Okay, so second statement is also false. Third statement, hydrogen bonding guarantees the complementarity of the strand. That is true. We just now wrote a complementary strand for the given strand of DNA. So that is because of the hydrogen bonding only. So A pairs with T, G pairs with C. So you will always have a complementary strand for a given strand of DNA. Then comes fourth statement, that is stacking interactions are sufficient to overcome the electrostatic repulsions of the phosphate. That is wrong. Both stacking and base pair hydrogen bonding interactions are required. So stacking interactions are not sufficient to overcome the electrostatic repulsions that are there in the DNA molecule. So from where this electrostatic repulsion is coming? So we have the phosphate groups protruding out of the molecule. I told you the bases are on the plane, and then the sugar phosphate backbone is protruding out of the molecule, so or out of the plane. So the phosphates groups, so phosphates are always charged, PO4 3 minus. So the charged groups, so now we have two negatively charged groups alongside each other. So they will have repulsion, both are negatively charged, so they will repel each other, right? They will repel each other. So there will be an electrostatic repulsion between these groups. Okay, so all the three statements, first, second and fourth, are false. Only the third statements, that is hydrogen bonding guarantees the complementarity of the strand, that is a true statement. Okay, this is clear? Is this clear to everybody? Okay, moving to the next question. The dash in the ribonucleotides renders RNA molecules susceptible to the strand cleavage in dash solutions. So we have two blanks here. The dash in ribonucleotides renders RNA molecules susceptible to cleavage in dash solutions. So what group in ribonucleotides leads to the cleavage of RNA molecules? We talked about hydrolysis, RNA hydrolysis. So tell me which group is susceptible, like which groups makes the RNA molecule susceptible to hydrolysis and under what conditions? Is it acidic or alkali? Okay, option A. Neeta says option A. Let's see. Is it option A? Yes. Option A is the correct answer. So remember, in the ribose sugar, I told you this was the first carbon to which the base is attached. The second carbon to which an OH group is attached. Third carbon also has a O, and then the phosphate or the rest of the RNA molecule. Fourth carbon, and then the fifth carbon is attached to a phosphate. This is the fifth carbon. So this second carbon that is attached to the OH, this OH actually attacks this. Under alkali condition, this hydrogen will get out of the system, which will lead to, so OH, if it gets deprotonated, it will become O minus, and then this O minus can attack the phosphate group that is attached from the next. So here another sugar molecule will be there, right? So there will be a carbon and then the phosphate. So another sugar molecule, another base, and then another OH, right? So this O minus will attack, and then there will be a breakage in the RNA molecule. That is the hydrolysis of the RNA molecule. So the, let's say we had a strand of RNA molecule like this. Okay, so let's say the hydrolysis is happening here. So the bond between A and C, the phosphodiester bond, this will break under alkali conditions. Why? Because under alkali conditions, under basic conditions, this OH can get deprotonated, and then this deprotonated OH basically will become O minus, and then this O minus can attack this phosphodiester bond. Okay, so 2' OH is making the RNA molecule susceptible to cleavage under alkali conditions. Okay, moving to the next question. Which among the following statement is or are false about DNA melting temperature? This is the TM. First statement is higher the salt, higher the TM. Second statement is higher the GC content, lower the TM. Third statement is low or high pH decreases the TM. So which statements are false? Which statements are false here? Okay, Neeta says option B. Okay, let's see. Option B, that is second statement is false. Okay, so which we had first statement higher salt, higher TM. Second statement is higher the GC content, lower TM, and third statement is lower high pH decreases the TM. So yes, the second statement is wrong. Higher the GC content, it will become, the TM will go higher up. Why? Because what is the melting temperature? Melting temperature is basically the temperature at which the double stranded DNA, I told you the DNA is double stranded, 5 prime to 3 prime, one strand, and then 3 prime to 5 prime other strand. This will become single stranded DNA. So 5 prime to 3 prime and the other strand 3 prime to 5 prime. Okay, so the breaking of this strand. So now you remember G had 3 hydrogen bonds with C, right? Sorry. So G had 3 hydrogen bonds with C, sorry. G had 3 hydrogen bonds with C. So to break GC pair, it will take more energy, right? It is easier to break 2 hydrogen bonds than to break 3 hydrogen bonds. If I give you 3 sticks to break together and if I give you 2 sticks to break together, it would be easier for you to break 2 sticks together, right? So that is why more energy means higher temperature is required to separate these two DNA if the GC content is higher. So higher the GC content, higher will be the TM. Higher the salt, higher will be the TM. Why? Because the salts will basically form like salts will have an electrostatic interaction with the negatively charged DNA molecule, and then it will make it more stable. It will make it difficult for melting. So if it is more stable, it will require again a higher energy to separate into two strands. Low or high pH can decrease the TM, that is true, because at a very low temperature or at a very high temperature the molecule will become unstable and then it will be easier for it to separate apart in two different strands. So it will require a lesser TM. So so correct answer is option B. So this is the TM curve basically. So this is the double stranded part and this is the single stranded part. So here this is the double strand and this is the single strand. So the temperature, as we increase the temperature, the double strand slowly starts getting converted into single stranded molecules, and the temperature at which 50% of the molecules are double stranded and 50% of the molecules are single stranded, that particular temperature, basically the midpoint of the linear curve, is the melting temperature for that particular DNA molecule. Okay, this is clear? So moving to the next question, question number 16. The overall rate of DNA renaturation is determined by, first statement is concentration of DNA, second statement is average length of DNA segments, and third is complexity of DNA. So overall rate of DNA renaturation. Now earlier we were talking about denaturation, that is separation of double stranded DNA into two single strands. Renaturation means joining the single strands back to make it double stranded, that means reformation of the hydrogen bonds. So overall rate of DNA renaturation is determined by the concentration of DNA, length of DNA segments, and complexity of DNA. So what would be the correct answer? All statements are correct. Okay, Anita says option A. Okay, the correct answer is option C. Actually all statements are correct. So higher the concentration of DNA, it will affect the overall rate of the DNA renaturation, because more the concentration of DNA, more time it will take to join together. More the longer the DNA length, if it is just two small fragments, it will join faster. If it is two long sticks, to join together those two long sticks, you will have to make hydrogen bonds between each and every base pair. If there are only 4 bases in a DNA molecule, only 4 pairings you will have to do. So there will be less number of total number of hydrogen bonds will be less. If there are 14 bases in a DNA molecule, it's basically you will need to make 14 base pairs, so more number of hydrogen bonds. So length of DNA segment will definitely affect the rate of the DNA renaturation. Then complexity of DNA. Complexity of DNA means what are the percentage of AT and GC content. So if GC content is higher, again the number of hydrogen bonds will increase, so the rate will differ. So if I have two different DNA molecules, one having 20% of GC content and one having 80% of GC content, both have equal number of bases, the rate of renaturation will be higher in case of lesser GC content, because it will be easier to, the renaturation will be faster, okay. And in case of higher GC content, the renaturation will be slower because more number of hydrogen bonds have to be made, okay. So concentration, length and complexity, everything determines the rate of the DNA renaturation. This is clear? So next question, because DNA is a highly charged polyanion, its stability to heat denaturation, option A, does not depend on hydrophobic interactions, option B, increases with increasing salt, option C, is independent of GC content, and option D, decreases with increasing salt. So what would be the correct answer here? We have already discussed this similar question. So what would be the correct answer? Okay, option C is independent of GC content. Okay, Neeta, do you think it is independent of GC content? I just told you the importance of GC content. Yeah. So the denaturation and renaturation is never independent of GC content, because GC content actually is the most important factor that determines the melting temperature of the DNA, the stability of DNA. More the GC content, the DNA molecule will be more and more stable, because the hydrogen bond is number of hydrogen bonds are more, okay. Option B, yes, option B is the correct answer, increasing with increasing salt. Sorry. Yeah. So because DNA is highly charged polyanion, its stability to heat denaturation increases with increasing salt. So if I have a higher salt content, the melting temperature will be higher, okay, because this DNA will be more stable. Why it is called a polyanion? Where is the negative charge coming from? Any idea where is the negative charge coming from? Yes, the phosphates. I told you the phosphates are giving the DNA molecule a negative charge. So there are multiple phosphate groups, each nucleotide has its own phosphate, and DNA is basically a polymer of nucleotides. So that it will have multiple phosphate, thus giving multiple negative charge. So polyanion. So multiple negative charge, and these negative charges are stabilized by the positive cations of the salt, and thus the total molecule becomes more stable and less prone to denaturation. So higher salt content means higher melting temperature, means the stability of any DNA molecule increases with the increasing salt concentration, okay. So option B is the correct answer. Next question, nucleoside is a pyrimidine or purine base covalently, option A, covalently bonded to a sugar, option B, ionically bonded to a sugar, option C, hydrogen bonded to a sugar, and option D, none of the above. So what is a nucleoside? We have discussed nucleobase, nucleoside and nucleotide. So in a nucleoside, what is the pyrimidine or purine base covalently bonded, ionically bonded, or hydrogen bonded to a sugar, or it is not bonded to a sugar at all? Okay, Neeta says option C. Anyone else? Okay. Hydrogen bonding to sugar. So, if you remember the structure of a nucleoside, it was basically, so you had a sugar. So, this is a ribose or a deoxyribose sugar. So, this is the 2 prime, 3 prime. Let's say this is a deoxyribose sugar. So, we'll have a phosphate. So, this carbon will be attached to a phosphate, and here you will have the base. So, this is one nucleotide. If I now remove the phosphate, it will be a nucleoside. So, nucleotide and nucleoside has the difference in, just a second. So, if I now erase this phosphate, it's a nucleoside. So, in this nucleoside, this base is attached to this sugar. I told you what is this bond called? It's called a beta glycosidic linkage. So, it's a covalent bond. It's not a hydrogen bond. The hydrogen bond is within the bases. So, this is one nucleoside. Another nucleoside will be there. So, when two nucleotides are pairing together, the bond between the bases is the hydrogen bond. What is hydrogen bond? Hydrogen bond is basically the bond between hydrogen and an electronegative atom. So, generally the bond of hydrogen with oxygen or bond of hydrogen with nitrogen, this is a hydrogen bond. Okay. Rest all the bonds between carbon and oxygen, carbon and carbon, then this bond, all these bonds are called covalent bonds. Okay. So, this is also a covalent bond. Here the bond is between carbon and nitrogen, and it's a bond between a sugar and an atom. So, this is a glycosidic bond. So, this is a covalent bond where it is called a glycosidic bond. Glycosidic bond means any bond that is formed with a sugar molecule is a glycosidic bond. Okay. So, if the bond was with sugar and like carbon and oxygen, it would have been an O glycosidic bond. In this case, it is called an N glycosidic bond. Okay. So, option A, yes, option A, covalently bonded to sugar. This is the correct answer. Okay. Hydrogen bond is between the bases. So, the bonds between A and T or G and C, these are hydrogen bond. Okay. Covalent bond is the strongest bond that is possible. It is stronger than the hydrogen bond. Okay. Hydrogen bond is weak. So, when you are heating the molecule, when you are heating the DNA molecule, it is getting denatured into two different strands, but the strands are not breaking into pieces. Why? Because the strand is attached with these covalent bonds. So, to break these covalent bonds, more energy is required, but to break hydrogen bonds, lesser energy is required. Okay. So, hydrogen bond is a weaker bond. Covalent bond is a stronger bond. What is ionic bond? Ionic bond is basically between any two ions. So, if a negatively charged ion is making a bond with a positively charged ion is making a bond with a negatively charged ion, this is ionic bond. So, the bond between NaCl, any salt and that matter, these are ionic bonds. Okay. So, the correct answer is covalent bond. So, nucleoside is a base that is covalently bonded to a sugar. So next question, question number 19. Which pyrimidine base contains an amino group at carbon 4? Option A cytosine, option B thymine, option C uracil, option D adenine. So which pyrimidine base contains an amino group at carbon 4? So you remember pyrimidine and purine. So purines had 2 rings and pyrimidines had a single ring. And which of the following is not a pyrimidine? First you have to find that out. Okay. Akshara says cytosine is the answer. Okay, let's see. Yes, cytosine is the answer. So adenine here is a purine, so adenine is definitely not the answer. Among uracil, thymine and cytosine, so we see the structure. So this is actually important. Like in proteins, it is important to remember the amino acid structures, at least the side chains. So similarly in DNA, you should remember the nucleobase structures. So you have here all these, the basic structure for all these are pyrimidine ring. So this is a single ring, pyrimidine. In uracil and thymine, the only difference is the presence of this methyl group at the 5th carbon. So in uracil there is no methyl group, in thymine there is a methyl group. In cytosine, instead of that methyl group, we have an amino group at the 4th carbon. So in both uracil and thymine, we have an carboxyl oxygen at the 4th carbon, but in case of cytosine, we have a amino group at the 4th carbon. So pyrimidine base that is containing an amino group at the 4th carbon, that is cytosine. Okay, so these are the structures of uracil, thymine and cytosine, 3 pyrimidines that are there. So try to remember as much as possible these structures. If you remember the structure of these bases, you can easily make a DNA molecule if I ask you to make. Okay, so you know how a sugar looks like. Now if you know how a base looks like, and now you definitely know how a phosphate looks like, so you can join these together. You don't have to mug it up. You can just understand how the different bases are forming and how the different molecules of DNA is attached to each other. So that's how you will understand these structures. You don't have to mug it up. So you see, if you understand the basic structure of pyrimidine, it's just the difference in 1 or 2 carbon side chains that are making it different. Okay, once you remember the structure of uracil, thymine and cytosine, if I ask you to make a DNA molecule with these 3 bases, you can easily make. Okay, so similarly, we will see the structure of purines also. Okay, next question. Thymidine can participate in hydrophobic interactions due to its methyl group. Thymidine is replaced by uracil in RNA. Thymidine normally forms 2 hydrogen bonds with adenosine, and option D, all of the above. What would be the correct answer here? So you saw the structure of thymidine. What would be the correct answer? Come on. Option D. Yeah, option D, all of the above. Yes. So thymidine, you saw the structure of thymidine here. It has that methyl group. Here it has that methyl group. So that methyl group can participate in hydrophobic interaction. It is replaced by uracil in RNA that we have discussed. It forms 2 hydrogen bonds with adenosine, so that also we have discussed. So all of the above statements are true for thymidine. Okay. Okay, question number 21. A purine with an amine group on the 6th carbon is option A adenine, option B cytosine, option C thymine, and option D guanine. So a purine with an amine group on the 6th carbon. So first of all, identify the purines and then answer. Okay, Akshara has answered adenine. Okay, so that is correct answer, option A adenine. So here we have the structures of purine. So we saw the structures of pyrimidine with single ring. For purines we have 2 rings fused together, 6 carbon and 5 carbon rings to fuse together, and in adenine we have an amino group at the 6th carbon, and for guanine we have an amino group at the 2nd carbon and a carboxylic oxygen at the 6th carbon. Okay, so the difference between these two purines is just on the side chain. So if you remember the structure of the purine, like the basic structure of purine, you can easily make these two structures out of the purine ring. So you can draw the purine ring and then place a amino group at the 6th carbon, it will become adenine. Place a oxygen double bonded to the 6th carbon and then an amino group at the 2nd carbon, it will become guanine. Okay. So it's not very difficult if you remember the basic structure of a purine and a pyrimidine. It's not actually very difficult for you to remember the structures of all the 5 bases that are there: adenine, guanine, cytosine, thymine, and uracil. Okay, so try to remember the structures of all the 5 bases. Okay. So moving to the next question. Question number 22. If a strand of DNA is 10% adenine, then what percentage of guanine will it possess? So option A 40%, option B 30%, option C 20%, option D 10%. So how to calculate this? If I have a DNA strand with 10% adenine, what would be the percentage of guanine? Sorry, Neeta, can you type? Your voice is not clear. I wanted to know what is the polarity and solubility of polar components in water. So, any charged species or even uncharged species sometimes can have polarity amongst themselves. So, that polarity comes from the difference in the electronegativity of 2 atoms. So, if I have, just a second, I'll explain here only. So, if I have an atom A bonded to atom B, okay, and let's say A is more electronegative, so what will happen? A will start so A is more electronegative, so it will start repelling the electrons and B will start withdrawing the electrons. So what will happen? There will be an partial negative and partial positive charge that will develop, and that will give a pseudo polarity to the molecule. So there will be a positive and a negative pole. Even if it is an uncharged species, due to the difference in the electronegativity, one molecule will withdraw the electrons from the other molecule, and because of that one molecule will become negatively charged and one molecule will become positively charged. Okay, and that's how there will be a difference in the potential of the whole system, and there will be a charge distribution, an uneven charge distribution that can lead to poles. Okay, so that's what we mean by polarity. Now what happens is, if you see a water molecule is H2O, right? We know oxygen is very electronegative. So what happens is, there is a partial negative charge and a partial positive charge developed on the atoms. So oxygen will have negative charge and hydrogens will have positive charge. Now because of this thing, what will happen? There will be a pull of electrons from the oxygen towards the hydrogen, from the hydrogen towards the oxygen. Okay, because of that pull of electrons, there will be poles defined. Because of that pole, the net polarity of the molecule, so this molecule will have some amount of charge even if the total molecule is uncharged. Now because of this, what will happen? So any other molecule that is also having this kind of polarity, this will like to dissolve in this polar solvent, that is water. Okay, so all these charged amino acids, and in case of DNA the charged sugar phosphate backbone, they are hydrophilic. They are called hydrophilic because they like water or aqueous environment. Why do they like water or aqueous environment? Because they have the same polarity, or they are also polar, and it's always like if I have a nature, I will like to be with people who have the similar nature. Okay, so it's something like that. So a polar substance will always like to be in a polar environment. A non-polar substance, on the other hand, will like to be in a non-polar environment. If something is hydrophobic, it will like to be away from water. Okay, and if something is hydrophilic, it will like to be near water. Okay. Okay, so you are asking alcohol is polar means what it does. So alcohol, all the alcohols have a group called OH, right? So OH is, again, oxygen is there, it is electronegative, so it is kind of creating a partial negative and positive charges. So if I have a methanol, so CH3 group is attached to OH. Now CH3 will have a partial positive charge and OH will have a partial negative charge. What will be more compatible with water, acetone or alcohol? So both acetone and alcohol are actually polar molecules, so both will be miscible in water. Okay, acetone is also highly polar solvent. So acetone has a ketonic oxygen in it. So all these oxygen containing solvents, most of the oxygen containing solvents, they are polar because the oxygen can make hydrogen bonds with the water easily. Okay, oxygen, any highly electronegative atom, it can form hydrogen bonds with water. Anything that can form hydrogen bonds with water will be soluble in water. Okay, so these are all polar components and they will be soluble in water or any aqueous. So if I take a polar compound and I try to dissolve it in acetone, the chances are it will get dissolved in acetone because acetone is also polar. Now on the other hand, if I take a hydrophobic molecule, I take a fatty acid. Okay, you all have seen oil at home. Okay, so if you take oil and try to dissolve it in water, what happens? Does it get dissolved? Does oil gets dissolved in water? No, right? Why doesn't it get dissolved? Because now the oil particles has hydrophobic groups in it. Okay, hydrophobic chains in it. So if you remember the structure of fatty acid, I talked about it in the previous session. So fatty acids have alkyl chains, the long alkyl chains, just CH2 attached to each other. So what it does, it makes it hydrophobic. There are no oxygen to make hydrogen bonds in the water. So what happens, oil forms a droplet in a water because it does not want to be around water, so it makes a clump and it stays with its hydrophobic friends. So all the oil particles, all the oil molecules will come together and they will make a group of their own. Since they are hydrophobic, they will stay together and all the hydrophilic molecules will stay together. Okay, so whenever I say polar, means it will be soluble in water, it will be hydrophilic. Anything that is non-polar, it will be hydrophobic. So all the atoms, mostly if you see a molecule and if you have to just guess whether it's polar or non-polar, try to see if it has all the carbons attached to carbons and hydrogens. Mostly it will be non-polar, it will be hydrophobic. If it has oxygen molecules and nitrogen molecules, then it will make, so if it has a amino group, a hydroxyl group like NH2 or OH, these kind of groups will make it hydrophilic, because now these nitrogen or oxygen, they can make hydrogen bonds with water. They will give it a they will give the molecule a polarity. Why? Because they are electronegative, so they will withdraw the electron towards themselves and they will generate a partial charge in the species. So all the ionic species are already hydrophilic, so they can dissolve in water easily because they can dissociate into ions. All the non-ionic or the uncharged species, they cannot dissociate in water into ions, but they can dissolve in water because of this property. Okay. Okay, so we diverted from the topic. We'll come back to the question. The question was, if a strand of DNA possesses 10% adenine, what would be the percentage of guanine in that DNA? Okay, option A, 40%. Yeah, so that is correct answer. How to calculate this? So you have, if you have 10% of adenine, so I've told you adenine pairs with thymine. That means 10% of adenine will have 10%, like if a DNA molecule has 10% of adenine, that means it will have 10% of thymine. If it has 10% of thymine, that means the total amount of adenine and thymine is 20%. So out of 100%, 80 is 20%. That means GC is 80%. Okay, if you have now 80% of GC, now again equal number of G will pair with equal number of C. So if you have total 80% of GC, that means 40% is G, 40% is C. Okay, so this is very simple. Neeta says option D. Why, Neeta? Do you understand why it is 40%? So whatever the percentage of adenine will be, the same will be for thymine, and the total percentage of adenine plus thymine will be 100 minus the percentage of guanine and cytosine. Okay, yeah, okay. So percentage of G comes to 40% if adenine is 10%. Okay, so such type of questions can come, like these are very small numericals in this topic. Okay, so again, what would be the nucleotide sequence of the mRNA strand from the DNA strand given below? So this is the DNA strand that is given. So this is the DNA strand that is given. So what would be the mRNA strand? Now you have to pay attention. They have asked mRNA strand, not the DNA strand. So what would be the difference in different in mRNA? There will be a uracil, right? So tell me what would be the complementary strand? Come on, tell me what would be the complementary strand? So if I have a DNA 3' to 5' and I am asking an mRNA complementary, again we will have opposite to 3' we will have a 5'. Now 3' first base is A. So opposite to A, I'll write here only. So this will be 5'. Opposite to A, what will have? Option B. Yes. So opposite to A, we will have U. Opposite to T, we will have A. Then C will have G. Then G, A will have again U. So in DNA, we had A paired with T. For RNA, we will have A paired with U. So same thing will happen. Just instead of T, we will have U. Okay, so again G will have C, T will have A will have U, and C will have G, and this will be 3'. So we have option B where 5', then U A triple G, U double C, A U G G, and 3'. So yes, option B. So now you see option D also has the same sequence. What is the difference? The difference is in this priming. So you have to pay attention to what the number here is written. So if it is 3', the opposite will be 5'. If it is 5', the opposite will be 3'. So you have to pay attention on this also. Okay. So very good. So option B is the correct answer. Okay. Next question is question number 24. Which of the following is not a component of nucleic acid? Sugar, phosphate, tyrosine and adenine. Which of the following is not a component of nucleic acid? This is very simple. Yes, tyrosine. Very good. So tyrosine is a amino acid. We have studied in the previous sections of our discussions. Tyrosine is an amino acid, so it's not a part of nucleic acid. Nucleic acid is formed with nucleotides, and nucleotides are basically a sugar group attached with a base and a phosphate group. It makes up a nucleotide. Next question. What are the three major components that make up a nucleotide in DNA? We just discussed, so tell me. Option A, glucose, ribose, nitrogenous base. Option B, phosphate deoxyribose nitrogenous base. Option C, protein unit deoxyribose nitrogenous base, and option D, phosphate ribose nitrogenous base. What are the components of nucleotide in DNA? Neeta says option B, phosphate deoxyribose and nitrogenous base. Yes, that is correct. So you have to pay attention what is there in the question. In question it was DNA, so it will be phosphate deoxyribose and nitrogenous base. If it was RNA, it would have been phosphate ribose and nitrogenous base. So you have to pay attention in that also. Okay, so one small change in a letter or a word can make a huge difference in this context. So just be attentive when you are solving these kind of MCQ questions for any exam, for any other competitive exams also. Read the questions carefully, okay? Question number 26. A section of mRNA is composed of 28% guanine bases. What percentage of RNA strand is cytosine bases? Option A 28%, option B more information is needed in order to answer this question, option C 14%, and option D 36%. Okay, Neeta and Akshara both have answered option A. Okay, so now in this question you have to, again I told you have to pay attention. In the question it says mRNA. So in case of DNA we can apply that rule of base pairing because DNA is double stranded, right? But in case of mRNA, it is a single stranded molecule, so it does not follow the Watson-Crick base pairing. It's a single stranded DNA. So if I have told you that it has 28% G, it does not mean that it will have 28% cytosine. Okay, more information is needed in order to answer this question, because in a mRNA molecule there is a chance that it has 0% cytosine, because it's a single stranded molecule. For DNA we could answer this question because it was a double stranded molecule, so we knew if there is 20% of guanine, it has to have a 20% of cytosine as well to match to balance that base pairing overall. But in mRNA, since it's a single stranded, there is no base pairing, there is no Watson-Crick base pair rules that are applied to it. So that's why you cannot determine the percentage of the nucleotide bases based on the given percentage for an RNA strand. Okay. So mRNA is not a double helix like DNA. RNA is only one single strand of nucleotides. This means that we are unable to say that there are just as many cytosine bases as guanine bases, even though they would be able to form nucleotide pairs in DNA. For all we know, there could be 0 cytosine bases. So that's why more information is needed before we make a conclusion as to how many cytosine bases are there in RNA section. Is that clear? So that's why you cannot say that 28% of G means 28% of C. Next question, question number 27. Which of the following statements is correct about the differences between DNA and RNA? Option A, DNA is present as a single stranded molecule while RNA is double stranded. Option B, sugar molecule in RNA has one more hydroxyl group than the sugar molecule in DNA. Option C, RNA is synthesized from DNA during transcription, but DNA can never be synthesized from RNA. Option D, RNA contains the same bases as DNA except uracil is present instead of guanine. Okay. So which of the following statement is correct about the difference? Tell me which of the following statement is correct. Only one statement is correct here. Okay, Neeta says option B. Sugar molecule in RNA has one more hydroxyl group than the sugar molecule in DNA. Yes, that is correct answer. We know that the 2' OH is missing in case of DNA. In case of DNA, we have a 2' deoxyribose. In case of RNA, we have ribose with the 2' hydroxyl attached to it. Okay. DNA is option A is DNA is present as single stranded. This is wrong because RNA is present as single stranded generally and DNA is present as double stranded. Option C, RNA is synthesized from DNA during transcription, but DNA can never be synthesized from RNA. That is again false. In cases of virus replication, we have a process called reverse transcription where RNA can be synthesized, DNA can be synthesized from RNA. So there are some viruses. So all of you are now familiar with coronavirus. So that is a RNA virus. So the genome, like our genetic information is there inside the DNA. For some viruses, it is possible to have RNA as the genetic material. Okay, and coronavirus is one of the good examples of RNA viruses, and since they have RNA as the genetic material, for them to transcribe their information into a protein, they have to convert the RNA into DNA in order to infect other species like humans. So they use our machinery to make their own proteins. Okay, and our machine knows how to make protein from a DNA, because our machine follows the central dogma of biology, that is DNA to RNA to protein. So for them, for those viruses to use our machinery, what they do, they simply first reverse transcribe their RNA into DNA and then use our machines to make protein, make their own protein from DNA to RNA to protein again. Okay, so this is called reverse transcription, and this was the process behind the RT-PCR that was being done during COVID tests. You must be familiar with these terms, the COVID and the RT-PCR. So this is actually the principle behind it. So those were the RNA viruses. So the RNA had to be converted into DNA and then amplified to know the amount of DNA and whether it is present or not in case of patients. Okay. And then RNA contains the same bases as DNA, except uracil is present instead of guanine. That is wrong. It is present instead of thymine. So yeah, HIV as well. Yes, yes, HIV is also a RNA virus. Okay, so moving to the next question quickly. Question number 28. Which of these statements are true for a glycosidic bond? So I've talked about the glycosidic bond in a DNA, so you should be able to answer this. Option A, the bond between sugar and the nitrogenous base. Option B, bond between two sugar residues. Option C, bond between sugar and a phosphate. Option D, bond between base and a phosphate. So which is the glycosidic bond? Which is the glycosidic bond in DNA? Option A, yes, bond between the sugar and the nitrogenous base. So I've discussed this previously also. So this is, so we have the sugar, the base and the phosphate attached. So this is the bond between the sugar and the phosphate. This is the glycosidic bond. And what type of glycosidic bond this is? This is a N-glycosidic bond, and it is held in a beta configuration. So it is a beta glycosidic bond. Okay. Next question, primary structure of DNA and RNA proceeds in which direction? 3' to 5', 5' to 3', 4' to 6', 3' to 6'. This is also simple. In which direction the primary structure of DNA and RNA proceeds? Nita says option B. Option B, that is 5' to 3'. Yes, correct answer, 5' to 3'. So how do we name this 5' and 3'? Basically you have the sugar to which the phosphates are attached, right? So the first base that will be there, the first nucleotide that will be there in a DNA, it won't have the phosphate because there is no bonding that is happening. Phosphate is attached so that the phosphodiester bond can form between two nucleotides. So this is the first nucleotide. Here the 5' is free, then 5' goes to 3'. From the 3' OH the phosphodiester bond forms. Again this phosphate attaches to the 5'. So the strand goes from 5' to 3'. Now the opposite strand will go from 3' to 5'. So the 5' carbon will be free at this end where the 3' OH is free, and then the adenine will base pair with thymine and thymine will base pair with adenine and so on. So I hope this is clear. what happens in hyperchromicity? So this is the last question. What happens in hyperchromicity? When DNA is in bound form, there is a decrease in absorption of UV light. When DNA is in when DNA is in bound form, there is an increase in absorption of UV light. When DNA is in unbound form, it is more stable. When DNA is in unbound form, there is increase in absorption of UV light. So what happens in hyperchromicity? What is hyperchromicity? Anybody? So hyperchromicity is basically the phenomena where when DNA is in bound form, there will be a when DNA is in unbound form, there will be an increase in absorption. So this is the melting curve I showed you. So this is the y-axis is absorption at 260 nanometer, and the x-axis is the temperature. So this is the double stranded form, and as we increase the temperature, the double stranded starts denaturing into two single strands, right? So this is the double stranded DNA form, this is the single stranded DNA form, and you see the absorbance is increasing. So when the DNA is in double stranded form, the absorbance is lower. This is a hypochromic state. When the DNA is becoming single stranded, the absorbance is increasing. This is a hyperchromic state. So this is hyperchromicity. The effect is called hyperchromic effect. Why this is happening? Because of the base stacking. So when the DNA is in double stranded form, the bases are stacked upon each other, thus decreasing the absorbance. Now when the bases are free, when the strands have broken apart, so now there are two single strands, so the bases are free, so the absorbance increases because there is an absorbance of each base, each and individual bases. So that gives rise to a higher absorbance for single stranded DNA than double stranded DNA. Okay, so when DNA is in unbound form, there is an increase in absorption of UV light. Okay, so this was the last question. Any doubt in any of the questions? Okay, so if you have no doubt, we can end this session. If you have any form of doubt, you can ask me, or we can discuss it in next session also. It's fine. Okay. Okay, so thank you so much for joining, and I'll end the session now. Thank you. Bye bye.